College of Pharmacy, I, Gargi Jogi from MPharm Semester 2, Department of Pharmaceutics, invite you all to the webinar on Process of Innovation Development and Technology Readiness Level by Dr. Shrinivas Savale, sir. This webinar is organized by Institution Innovation Council, LM College of Pharmacy. I would like to invite Dr. Mamta Shah, ma'am, in charge principal, HOD, Department of Pharmacognosy, for a welcome address. Over to you, ma'am.
Thank you, Gargi. Good afternoon to one and all. As we know, importance of innovation, and our college, being a premier institute of pharmacy education, is involved in this direction in a great way. We at our college have Institute Innovation Council, SSIP, and even AIC, which is first of its kind allotted to our college as far as health sector is concerned. And what to say, we all are pharmacy discipline members, and we know what is the role of innovation in the drug discovery and overall life. So not taking much of your time, I welcome you all and hope that you will all learn and enjoy the talk from our CEO of AIC, Dr. Shrinivas Savale. So over to Dr. Savale.
Sure. Gargi, can I start or?
Yes, sir. I would like to introduce you to all our guests, if that's fine.
Sure, go ahead. Quick and brief.
Yes. Now I would like to introduce today's speaker, Dr. Shrinivas Savale, sir, CEO of Atal Incubation Center, LM College of Pharmacy. He is an acknowledged leader with over 22 years of professional experience in drug research, development, and compliance in pharmaceutical organization, CRO setup, and academia. Sir was associated with Torrent Pharmaceuticals Limited as General Manager, Bioevaluation, Clinigen International Limited as Head Bioanalytical Research, and Torrent Research Center as Scientist at Medicinal Chemistry Division. Currently, Sir is a member of Clinical Pharmacokinetic Pharmacodynamic Subcommittee of Biosimilar Focus Group. He has been a reviewer for various national and international journals, invited speaker at various national and international conferences, and has more than 30 publications, a book chapter, and many scientific presentations to his credit. With this, I would like to invite Sir to begin with this session. Over to you, sir.
Thank you so much, Gargi, for quick introduction. I'm sharing the screen and I'll start going. Just confirm if you can see the screen.
Yes, sir, it's there.
Okay. Yeah, so good afternoon, those who have shown interest in this topic, and you are here on a Saturday, that too at 4:30. The topic that I'm trying to discuss is process of innovation development and technology readiness level. Now, the moment we talk about innovation, I mean, this is a very big buzzword now. Everybody talks about innovations, and then next comes is people talk about startups and then unicorns and so on and so forth. So it is very important for us to understand what innovation is and how a specific innovation gets developed and comes to the market, and how it changes the entire, you can say, the ecosystem when it comes to bringing something novel which adds value, right? So let me start with the basic aspect, that is, or you can say one of the very unique traits, that is creativity. I'm just putting some cartoons here. Like if you look at this, there are several points, right? You can see many circles with different colors. So this is exactly what we have knowledge. These days we have plethora of information available. And that information, when you actually read and understand and it converts into knowledge, which one should apply as we move forward. And how to use this information or the knowledge, it comes with experience, right? So if you look at this second carton, you will see that the circles or the dots have been connected in a structured manner, and it gives you one particular structure out of this, you can say, chaos that is shown on the left side. And further, so this is obvious that as you get more and more experience, you know how to use the data, how to use the knowledge for bringing some meaning out of this all information. But then the inherent capability sometimes are to looking at the same information differently brings the trait that's called as creativity. These are the same dots, I mean, same information, but you are bringing something different, which is out of the box. So you think creatively. And believe me, the most important skill in the future will be the ability to connect the dots in your own way. So this is something I'm sure that each one of us should keep in mind, that our ability to connecting the dots and deriving some meaningful insight from the information is going to be the need of the hour and also need of the future. So when we look at the creativity and we want this creativity to be converted into innovation, and which ultimately brings new products, processes or services or businesses to the market. So it's not that easy, right? So when we say creativity, there are other traits, like creativity is, you can say, outcome of other traits, like you have certain skill set, curiosity, you are motivated, and you are imaginative about certain things in a specific manner, and that becomes your creative expression. But to convert the creativity into innovation, there is a first barrier, that is the teaching support, or you can say the mentoring. So you need actually right guidance at right time to convert your creative thoughts into innovative idea or innovation per se. And then that innovation, when you want to really translate it into a new product, the second important barrier comes is the economic environment or the conducive economic atmosphere, where the innovative thought or idea, you need to experiment, you need to check whether it is working the way you have hypothesized, and whether it will then convert into a product which the ultimate customer or the consumer would like to have. So as I said, so creativity translates into innovative ideas, they get translated into the innovation, which actually can translate into new product, process, or services. But then the moment you talk about any idea, be it a creative idea, you would always like to look at the uh, how best that idea should be, or how innovative that idea should be. So the typical benchmark criteria for any innovative idea is such idea should be helpful to the people, it should save time, it should be adding to convenience, and it should be cost effective, as well as it should improve quality of life. So either one or multiple of these benchmark criteria make that idea, innovative idea useful, right? And how does this process work? So the innovation process chart, if you look here, so the first and foremost important aspect is identifying a problem. So if you are really working on some specific problem, how clearly you identify that problem is important. So based on that, your approach to bring a solution for that problem is going to be derived. So once you have identified the problem, then you come up with ideas to solve that problem. So you try, it may not be just a single idea, but multiple ideas that you may have, right? So uh, it is like you have multiple routes to reach to LMCP from your house, right? So you have multiple options. So like that, one problem may have multiple solutions, but then each of these solutions may or may not work, right? So you need to evaluate this idea or the solution for the fitment and for the best possible solution in a given circumstance. So once you evaluate the idea for its credit, then you pursue this particular idea into developing it into whatever. It may be a new product or a process or even business model for that matter. So you start working on developing that innovation. That's what we will go in little detail as we move forward. So this is typically like the most of you have heard this terminology like development of prototype and prototype testing and validation and so on and so. So this innovation is then first used or validated in the kind of expected end users, or you can say expected end environment. And once it is found to be working as it was hypothesized and bringing all the value additions, then it is ready for commercialization. And even if it is like we talk about in the technology sector, if you have developed this technology which is now working, so this technology is now ready for transfer and taking it to the masses. But during the course of this entire process, we are generating a lot of knowledge, and either that knowledge can be protected in terms of protecting the intellectual property, or you would like to put this as open source information that anyone wants to use and build on this information. This knowledge can be diffused in the open source. So this is what the innovation process is, and it is very important to have empowerment through innovation if you want novel solutions for certain problems. So everything starts with something called as thinking. And now, most of us are also talking about quality by design, right? So we start looking right from the inception of maybe a formulation or a drug or an analytical method or anything of that sort. So everything starts with certain thoughts or some solution that we think may be feasible. And only thing is that is probably a madness. You have some random ideas or the solution about the solution, and you want to really bring some order to this madness. So that's where this design thinking process is very important. So what happens? What are the components of this process? So basically, there are 5 different components of design thinking process. First is empathize, then define, ideate, prototype, and test. So when you are actually thinking about the problem, means you are empathizing with that problem. So you learn about the audience for whom you are designing this particular solution. All right. So who are the end users, or who are those who are ultimately going to get impacted by your solution, right? So you empathize and then you define or construct a point of view which is based on the user needs and insights, right? For example, you see that most of the smartphones, they undergo a lot of variations every now and then, rather modifications every now and then, mainly because of the feedback that is coming from the users, right? So they, based on the user needs, they are further upgraded. So you define the solution and then ideate, that is, you actually brainstorm and try to come up with creative solutions for that particular problem. So that's where you may have one or multiple ideas, and you need to really figure out which one will work. And to check how it will work, you come up with a prototype, right? So, or it's like building a representative model or a representation of one or more of these ideas, which you want to show it to others, right? So this is very, very important stage that you arrive at a prototype. And this is something like, if I have to explain in terms of what we do at our innovation initiatives at LMCP. So at IIC, we are empathizing and defining an ideation probably. And then when it comes to building a prototype, that's where SSIP is supporting with some funds to really build that particular representative model. And then you can test that particular prototype by returning to the original user group or testing this idea and getting feedback from the so-called users that they would use your particular solution. So it is very important at the initial stage that you create to create any meaningful innovation. One should know the users and they should care about their lives or their requirements. This is very crucial first step. Once you have that, then you actually frame the right problem. Now, as most of us know that how important it is to actually have clarity on objective, right? Or clarity on what is the problem that we want to solve. If you know the problem correctly, then that is the only way to create right solution. So if your problem identification is not robust, then I think you are going to go like in different directions. And once you have correctly identified the problem, now you start working upon ideas, right? And it's not necessary that you should come up with only right idea, but it is important that you come up with as many ideas as possible at this stage. So what can be different solutions for the same problem? And based on that, you start like you build to think, that is the prototype that you start building, and test this prototype to learn, right? So certain things don't work the way we hypothesize or the way we conceptualize. So it is important that you have this prototype which is built and amended based on its testing, right? So that's where you go for testing of this prototype. And believe me, testing is the only opportunity when you are learning about your solution as well as the users who are going to use your solution. So this is called as design thinking process, and which is again a very important stage in innovation. So you are ideating a particular solution and then trying to bring it in the form of a prototype and testing. Now, once initially you test the prototype and you think that it is working, the next step comes is the validation. So it's important that anything that we do needs to be validated, cross-checked, and it has to reproduce the way we have hypothesized. So there are again certain steps when we talk about validating a prototype. So first is the user should understand the concept. So when you most of the times when we come up with a solution, you reach out to probable users and take their feedback. For example, I am coming up with a face recognition app. You talk to people and try and find out what is their feedback, whether people will be using this kind of app or they think this is crazy idea, useless idea, whatever. So users should understand what is the concept, right? And the triggers for such things are always like there are many triggers which are internal or external triggers, and users are actually looking for certain solutions that we would have thought of. So this is the first validation in terms of whether whatever solution you are proposing, whether the user is understanding or not. Next comes is, yes, now they have understood what solution you are offering, but would they like to use it? That is the next question. So what is the motivation for the users to use your particular solution, be it a product, process, anything, business idea, anything of that sort? So it is important that the user should find it very user friendly, as we call it. So they should not have a lot of difficulties, or they need not have to change their habits or processes which they are accustomed to too much. So that comes the second validation. Next comes is, okay, now your concept is clear, people are ready to use your product, but does it add value? That is important. So what is that value increment is happening because of our innovation? That is the third validation. So basically one has to look at the concept presents certain rewards which are satisfactory to the users, and they continue using this prototype. So it's not only one-time use. You should look at, will they be using it again and again? So your product or whatever solution you have built, it is acceptable to the users or the customers, and not only once, but they will continue using your particular product. Now, this all information you are getting at the prototype stage is called as prototype validation. And this is where you actually build something called as a minimum viable product. So this minimum viable product has already been tested for clarity on concept, usability, and value addition. And this is the time when you can do as many experimentations as possible, because this will help you to understand if any changes are to be made to the product or prototype that you have made. So one has to be very cautious while doing this MVP testing. Is when you are, most of the times we are exploring look and feel of the solution, but you should not lose focus on the functionality and usability of the solution. That is what is kind of a wise advice, you can say, that you definitely have to keep focusing on the functionality and usability of the solution, and look and feel are actually add-ons. So it is, you may have many ideas at the beginning, right? But you focus on only probably top 3 ideas which you would like to convert into design or the prototype. So make initial prototype and validate these prototypes. And prototype validation is again not a one-time job. It's like it's iterative process. So you build particular, for example, as I will give you some examples that we do, right? For example, formulation development. We are making certain batches. We look at the desirable properties if we are getting or not. If not, you are modifying the formulation, right? And again, testing it. So it is every time you have iterative process of validating that particular prototype. So ultimately, in this entire innovation development, what is called as a loop, which is build, validate, and learn feedback loop. This is always working. So you build a prototype, you validate it, learn from during the validation process, what are the feedbacks, positive, negative, both ways, and again, bring those learnings back to the prototype and build the more improved prototype to go to the next level. Now, so when you are doing this, the last step of validation is actually testing with the real users, right? So you need to really look at the most trusted way of validating this prototype, because, like for example, I'll give you example of even clinical studies that we do, right? For anti-cancer drugs, we are actually testing them in cancer patients, right? Those are the target disease population, right? So you look at for whom you have built this solution, who would be your real users, and test your prototype with them. During this process, one has to observe the user behavior and understand their reactions to the prototype, right? So this is very important. And more observant you are, you may get more insights and you can build a robust product or the process. So while validating, you should create a test plan with tasks, right? So you should have a complete list of what all tests you are going to do. And you should, if you are doing a questionnaire based validation or feedback mechanism, you should ask open-ended questions and broad questions related to product concept. And you should not ask leading questions, like you should not hint them about the answer for the question. So this is first step. Next is you need to look at the sampling the right users, right? So there should not be outlier opinion popping up after your survey, for that matter. And then when you're conducting the test, there may be multiple ways. You can do it remotely or you can do in person. And you can also engage facilitators who has in-depth understanding about the user, right? So if the users are not giving the right feedback, probably this facilitator can help you to get the right feedback from the users, or also devise the right strategy to get this information. Further, you actually need to ask users to think loudly and give whatever feedback they think is about that particular solution, right? And it is important sometimes that you also engage some cross-function observers to monitor the user reactions. So uh, if I have to say that you are talking some independent intervention for validating your prototype. Now, we have been talking about the innovation process, innovation development, but I thought let me also give some perspective about what innovation itself is, right? And the moment we talk about innovation, most of the times we like we want to discriminate, invention and innovation, right? So simply putting, invention is creation of the product or introducing of a process for the first time, while innovation is improvement on or a significant value addition to existing product, process, or service. So this is the difference between invention and innovation. And so innovation is always you have something you are doing differently, but you are doing incremental value addition. So innovation is the change which creates or adds value and provides you competitive advantage. So this can be new product, production method, markets, new forms of organization, business models, and so on and so forth. So broadly, innovation is of two types: demand pull innovation and technology push innovation. So demand pull is where you already have the market and the market tells you what they need. And that's is demand pull innovation. So based on the market's requirements, so we are generating incremental innovation in the product, process, or services, and so on and so forth. So you need not create new markets. If I have to give some examples, Uber, Ola, Zomato's, even 1mg Pharmacy, these are like demand pull innovation. So technology was already existing. The information was, if I had to just give example of transportation, Uber and Ola, we already have transportation systems, right? And what advantage Uber and Ola is bringing is convenience to your doorstep or wherever you are, right? So that is like, so technology was available, cars were available, drivers were there. They just created an aggregator platform and brought the convenience to us. While on the contrary, technology push innovation is something which is hard on, which is heavy on R&D, and it brings new technologies and it generates actually more radical innovations, as well as it has to generate its own market. For example, you talk about Facebook. This was like first social networking, you can say, solution that has come in. 3D printing, wearables. We talk about gene and cell therapy. These are like novel modalities of therapy which are coming. They are really heavy on R&D and they are pushed by technological innovations. So once we understand what is invention, innovation, types of innovations, then it is very easy to relate how the innovation is developed, right? And when we talk about life science, especially the pharma and healthcare sector, we see that there have been a lot of innovations happening. Like this is a PCT filings data, that is number of patents published versus year here. So you can see that from 2000 to 2018, you have like almost, you can say here, 3 times the patents got published as far as medical technology is concerned. And something similar you can see like more than 2 times in patents have been published for pharmaceuticals, and similar ratio for biotechnology field. And as we saw from 2020 with the pandemic also, there is significant contribution of new technologies or new innovations that are coming in this field of pharmaceuticals, biotechnology, organic chemistry, or even medical technology. So you see here, there has been an increment in the number of patents that have been filed in these areas. So as we see that there is a lot of thrust globally on innovation, and more innovations are getting published and patented. And in this context, there is something called as Innovation Index, so Global Innovation Index, which was published by WIPO. And in Global Innovation Index 2021, there were 130 economies that participated, where each economy was rated based on the innovation input and innovation output. So in 2021, you may wonder it is not USA, but Switzerland is holding the top position now for many years. And India has stood at 46th position. And India's performance, if you look from 2015, we were at 81st position. And I'm showing here, in 2021, we are at 46th position. So there has been tremendous improvement in last 6-7 years as far as innovation inputs and outputs from India are concerned. And as you can see here, in the Central and Southern Asia, India is on the top as far as the innovation economy is concerned. Similarly, Niti Aayog has come up with India Innovation Index to look at how the innovation ecosystem is flourishing in India, and they had ranked different states and union territories here. You can see here that in Gujarat stands at the 8th position as compared, and Karnataka is on the top. And in 2019, Gujarat was on the 9th position. So there has been some improvement in ranking within India. And now, the moment we're talking about innovation, there are several aspects or several factors. So there are many key stakeholders and enablers in any innovation ecosystem. So here I'm just focusing on the pharma and healthcare innovation ecosystem. So you'll see that on the right side, the big pharmas and biopharmas, or even startups and entrepreneurs, are contributing a lot in this innovation ecosystem of pharma and healthcare. Even academic institutes and their collaborations are helping a lot to come up with newer and newer innovations. And but natural clinical research institutions and hospitals are one of the key stakeholders in this field. And to foster the innovation, there is a need of adequate infrastructure, financing or funding, and as well as there has to be supportive regulations and policies, which we have started seeing that we have now some of the schemes opening up even in the pharma and healthcare sector, which is encouraging innovations to come forward. So when we're talking about innovation, there is a systematic way of looking at how ready is this innovation or technology for commercialization. So when we talk from idea to market, so technology readiness level is nothing but it is a measure which estimates the technology's maturity of core technologies in the program during the selection process and in subsequent monitoring and evaluation phases until these technologies or the products utilizing them attain the market readiness. So this is the definition. Now, let me simplify, give you a little historical aspect of it. So this TRL scale is a nine, you can say, there are 9 steps in a TRL scale, which was originally introduced by NASA, conceived in 1974 and defined in 1989. So they have come up with a metric with 9 technology readiness levels for describing the maturity of a technology. So the lowest being TRL1, which is the ideation stage, while highest being TRL9, which is when the technology is ready to apply or ready for commercialization. So the in-between levels, between like level 1 and level 9, they cover this establishing the proof of concept, developing the prototype, doing the functional validations for the models, or even clearing the mandatory regulatory barriers uh, between the levels towards market introduction of these technologies and products. So basically, technology readiness level is just a metric which tells you at what stage the specific innovation is from their journey from idea to market. So I'm just showing a small video just to simplify this technology readiness levels. So bear with me for just a second. And
Technology Readiness Level, TRL. Technology readiness levels are a method of estimating a technology's maturity. So, what are the TRLs? TRL 1. The basic principles are observed and reported. To help you understand in simple terms, I'll use the example of a tire. So one day you're playing around in a lab and it hits you. You come up with a new synthetic rubber compound. You report it and start investigating. Well, what's next?
Hello.
TRL 2.
Sir,
This is
video
where
is not
invention
visible.
begins.
sorry,
The basic principles have been observed, so
Sir,
practical
video is not
applications can be
visible.
performed.
It is only audible. So it is just
In the case
audio.
of the rubber, this rubber
yeah,
could be
give me
used
a
for vehicles'
second. I'll
tires.
Oh, no
TRL
problem, sir.
3 is the proof of concept. Analytical and laboratory studies are required to see if the technology is viable.
give me a second. I'll play it as a hyperlink. I had
Sir, no problem,
introduced
sir.
hyperlink.
My job is to make college easier because students have a lot on their. Sorry, coach.
Technology readiness level, TRL. Technology readiness levels
Can you
are a
see it
method
now?
of estimating a technology's maturity.
Yes, sir. Yes, sir. It's visible. Thank you, sir.
TRL 1. The basic principles are observed and reported. To help you understand in simple terms, I'll use the example of a tire. So one day you're playing around in a lab and it hits you. You come up with a new synthetic rubber compound. You report it and start investigating. Well, what's next? TRL 2. This is where invention begins. The basic principles have been observed, so practical applications can be formulated. In the case of the rubber, this rubber could be used for vehicles' tires. TRL 3 is the proof of concept. Analytical and laboratory studies are required to see if the technology is viable. The rubber is tested for strength and forming capabilities to prove it's suitable to be made into a tire. TRL 4, laboratory testing of a prototype component. Here, you test the rubber to see if it has appropriate wear characteristics. TRL 5, laboratory testing of the integrated system. The rubber is used to construct a prototype tire and tested on a wheel. TRL 6, a prototype system demonstrated in an operational environment. The tire is mounted on a wheel and tested on a rolling road. TRL-7, the design is virtually complete and tested in a real environment. At this point, the tire is tested on a vehicle on the road. TRL-8, proven to work under its expected conditions and certified to be used. The certification will allow the tire to be used on the road. TRL-9, the system has successfully proven to function and is ready for commercial deployment. The tire has been tested through a life cycle to ensure it is safe for use. Above TRL-9 is introduction to the market. The product is launched commercially. These are the technology readiness levels brought to you by Warren Barth. For more information, check out the description below. Thank you for watching.
Yeah, so I'm just
નમસ્કાર listeners ને બધાને ખૂબ ખૂબ હાર્દિક
sorry. So this
અભિનંદન
is
છે. મારું નામ હેમંત પરીખ છે. ગોપાલ પાઠશાલા સિટી સેન્ટર અમારો આશ્રમ રોડ પર પ્રોજેક્ટ ચાલી રહ્યો છે. નારાયણ ગ્રુપ વતી આપ સૌનું
yeah, I'm so sorry. I just couldn't close it in time, but can you see the screen now?
Yes, sir.
The slides are visible.
Okay. So I think that gives some sense of what the TRL is, giving a very simple example. And then I really would like to now shift gears and take these TRLs to our life. In the sense, we talk about TRLs for drugs, devices. We look at what are the TRL levels for vaccines or biosimilars and other stuff. So basically, I'm sure most of us are familiar. And now, if we have to link the drug discovery and development process, so now you can link it with TRLs, right? So the basic research is more like ideation. And then when you get FDA approval for getting in the market, that's your TRL-9. And in this entire journey of TRL-1 to 9, we of course do a lot of, like, we come up with a prototype, you can say molecule, and that we are testing here, whether it works in preclinical models or not. And then if yes, then we are taking it into clinical studies where we do phase 0 studies with subtherapeutic doses to look at the pharmacokinetics of the drug, and subsequently look at phase 1 trials and look at the initial safety testing in small group of healthy volunteers. And then we really go to the patient population actually and look at in small group of patients whether this particular prototype drug or formulation is working or not, right? And then this is further can be in terms of looking at proof of the concept, that is TRL-4 that we are discussing, and then do sometimes dose range studies at this particular level. And then we go for actually validating or confirming the safety and efficacy in large number of patients. And that's when actually you actually have tested now it in the target population and the drug becomes ready for commercialization. So to your TRL-8 and TRL-7, and then the review becomes TRL-8, and ultimately you go to the market, that is TRL-9. Now, let me walk you through some of these steps for individual classes. Like if I had to apply this TRL for drugs, including the delivery systems. So ideation is when you identify the need of a particular drug and look at the basic principles, and you would have done scientific research, which is then converts into applied research and development. So that's your TRL-1. And TRL-2 is the proof of principle, where this idea gets developed, your hypothesis is tested, and you develop the protocols. So where you are doing initial level of in vitro studies or biochemical studies to just check proof of principle. And then the proof of concept needs to be demonstrated. So you are doing this hypothesis testing, right? Where you are doing limited number of in vitro models and limited in vivo efficacy studies, which are sometimes proven by in vitro model studies, right? Maybe cell-based models or ex vivo experiments, we have organoid cell models, so on and so forth. Or this is also in vivo efficacy in small number of animals. So that becomes TRL3, where you are having proof of concept demonstration. And the POC gets established when efficacy and safety of candidate formulation is demonstrated in defined animal model. So that is the TRL4 where POC is established. And then comes the early stage validation where we are, where we are doing preclinical studies, including GLP efficacy, acute and chronic toxicity studies, and we generate sufficient data for DCGI submission to get approval for clinical trials, right? And we get maybe if it is a NCE or NBE, you get approval for conducting a phase 1 trial. That's your TRL5. And TRL6 again is part of early stage validation, where you do GMP manufacturing of the investigational product and maybe especially for clinical trials. And your phase 1 clinical trials are completed, and now you submit this data to DCGI and seek approval for the phase 2 clinical trials. Then comes the TRL7 or the late stage validation, where phase 2 clinical trials are completed, and this data actually again is submitted and reviewed by DCGI, and then DCGI gives permission to conduct phase 3 clinical trials. And those are phase 3 clinical trials are the pre-commercialization activity or TRL8, where these trials are completed successfully, and then DCGI gives us approval for NDA and provides manufacturing license. And once you get this license, that's then comes the commercialization or post-market studies level, that is TRL9. Now, something similar, if you kind of go here for vaccines, right? So we have been hearing about vaccines a lot last 2-3 years. So in this case, again, TRL1 is again identifying the need, right? You do the basic research and understanding, develop understanding. But in proof of principle, you actually conduct epidemiological studies for vaccines, right? So based on that, your research idea gets developed, hypothesis is formulated, and you develop the protocols for doing in vitro studies or developing the working cell bank. And in TRL3, that is proof of concept demonstration, then you are actually doing the studies in limited number of in vitro models and also limited in vivo efficacy studies, which supports the formulation development, or it also involves completion of the in-house testing of the formulated vaccine by in vitro models as well as in vivo efficacy in limited number of animals. Same thing as we saw for drugs, when it comes to TRL4, you now need to check efficacy and safety here, especially for that vaccine candidate using the animal models. And then the results of the serological studies in different animals at preliminary level are done. Efficacy is defined in vivo models. And you have manufacturing and QC release of vaccines for studies as well as scale-up development. So your now proof of concept is established at this stage, and now you are ready to go to the next level, that is preclinical study. So again, early stage validation, typically preclinical studies, and they will the design and requirement of clinical studies will be defined based on what is the modality that we are testing, right? So you are also here looking at acute chronic toxicity and also repeat dose toxicity. And when it comes to vaccines, again, sometimes immunogenic potential is also verified as we go along. And TRL6 comes to based once you have finished your preclinical studies. In TRL6, you are looking at the GMP manufacturing of the material that is being used for clinical trial. So raised all TRLs are same as we saw for the drugs. So here in TRL 9, but we have post-marketing commitment and post-marketing surveillance. That is phase 4 clinical trials need to be done for the vaccine drug candidate. Now then, coming to biosimilars. So in case of biosimilars, again, the approach is little different. And as you say that you first look at what is the scientific knowledge base that is available when you are trying to develop a biosimilar. So that includes your patent status and assessment of different technologies that are being conceptualized. And as a proof of principle, you are looking at developing the hypothesis and experimental design. And here it is important that you identify the innovator product. Right? So in biosimilars, you always need to compare the proposed biological product and compare it with the reference or the innovator product. It is very important that you identify the innovator molecule or the product, and you need to develop the assays to test the activities of the candidate molecules both in vitro, and at this level you also look at the high expression clones are available for proceeding for manufacturing. At TRL3, you are actually identifying and characterizing the preliminary products. So as I said, you need to look at the comparative evaluation of the innovator product as well as the biosimilar in terms of physicochemical characterization as well as biological characterization in terms of activity in vitro as well as in vivo. And at this stage, you will be doing the cell line characterization as well as cell line characterization of the master cell bank as well as the working cell bank and process development. So at proof of concept demonstration, it is very important that you demonstrate the biosimilarity, both in terms of in vitro efficacy and preliminary in vivo efficacy in small animal models. And as you move up on the TRL, so at TRL4, when you are establishing the POC, you are working on developing the process, optimizing it, and demonstrating the biosimilarity. This is where you need to generate consistency data for different batches of formulations. And then you need to optimize the process. You would have done this process optimization for getting the material for preclinical studies. And you need to generate at least 3 consistent batches at this level. And this is the stage where most of the development work comes to, you can say, end. Like you would have developed the formulation, you would have finalized the route of administration. You also have prepared the draft product profile, and the process is optimized, and you get the approval for conducting for preclinical candidate from the relevant bodies. So as biologics, you also need to get approval from RCGM or GEAC, that is Genetic Engineering Approval Committee and Original Committee for Genetic Modifications. So when it comes to the early stage validation, in case of biosimilars, at TRL5, you continue with advanced characterization of this product and you initiate the manufacturing. So you would have also conducted preclinical studies, both in vivo toxicity and efficacy in relevant models. Not only that, you are also characterizing the biosimilar product with the PKPD studies, ADME characterization as well as immune response. This is very important for the biological molecules. And I mean, these are data are required for necessary regulatory filing. And if you are not manufacturing, probably you are identifying manufacturing partners and submitting the preclinical data to RCGM. So this is TRL5. And at TRL6, technology readiness level 6, you are initiating the regulated production and regulatory submission. So that is, you are looking at GMP compliant manufacturing, and study the stabilities of biosimilar. You develop assays, analytical methods for product characterization release. When it comes to late stage validation, that is TRL7, here you are working on the scale up. You also complete the process validation, GMP process validation, and look at the consistency, lot to lot consistency of manufacturing, and also head towards regulatory approval. So you have scalable and reproducible manufacturing process at this stage, right? And here, as you can see, you are actually going to phase 3 clinical trials. So you have complete stability studies of the GMP drug product, you have dosage form, you have container consistent, and both dosage form as well as container you would have studied, and you are looking at the consistent target product profile. So at TRL7, you are getting DCGI's approval to conduct phase 3 clinical trial. And this phase 3 clinical trials data is then submitted for approval or for licensure to regulatory body, and that is the pre-commercialization stage or TRL8, right? So you are submitting the biological licensing application here to the regulatory body, and once you get approval, you at TRL9 you go for commercial application, manufacturing license. Now this technology is fully developed and it can be distributed and marketed. And again, in biologics, you have a post-marketing commitment, so you continue with phase 4 clinical trials. These days, we are seeing that technology is impacting a lot in our sector, any sector for that matter. And there are TRLs even for the technologies like artificial intelligence, big data analytics, and so on and so forth. So here again, the levels are more or less like same 9 levels if I look at. So first level is again identification of the need, and you need to develop the basic use or properties of the software architecture, look at the mathematical formulations and general algorithms. So that is happening at ideation stage. At TRL2, actually research ideas get developed, and then this concept technology concept is formulated, and then you are doing some analytics studies and also start coding and comparing with the competing technology. So that is what happens when you want to prove the generate proof for the principle. And at TRL3, you are actually doing pre-alpha testing, or you have your script is getting ready and working draft is created. But at TRL4, now you are actually building the limited functionality environments and validate the properties and analytical predictions of this particular solution that you are trying to build. So here you also look at laboratory results showing validation of critical properties, right? So this is what happens at TRL4. At TRL5, again, early stage validation is TRL5 and 6. So these developed software technologies are supposed to be integrated with different aspects in existing systems. So that is being tested. Further, these technologies, when we want to implement, they should conform to the target environment and interfaces. So that is also needs to be checked at early stage validation. You are also conducting rigorous alpha testing, and at TRL6, you are then checking the feasibility of this technology at a full-scale realistic problem. So in case of technology, the readiness levels are different, right? So you are validating this technology in a relevant end-to-end environment, or also a rigorous beta testing is done at TRL6. So this is very important stage for software-based or technology-based solutions. And for them, the late stage validation is done by rigorous testing and validation by third party. The third-party validation has immense importance in case of these solutions. And when it comes to certifications, that is pre-commercialization, you need to check the quality of the software by some of the international standards such as ISO IEC 9126. And you also need to abide by or comply with the data privacy and protection as per the international standards such as HIPAA norms, right? And once you are done, then you can launch the software. So, and after launching the software, now here you will see that launch of software is commercialization actually. But then at TRL 9, you continue improving your algorithms, your software, your tool, your technology. And sorry, so those improvements can be to meet the needs of the customer, or those may be again, you want to really add new features over and above what are available in the solution. And also I wanted to touch base upon the veterinary products as I thought I will give overview in terms of our sector, life sciences, especially pharma and healthcare. So we also have veterinary applications and veterinary products. So that's where you look here at, again, the moment I say ideation, it is need identification, right? So you look at the basic principles, look at what is the status of science around that. And then you develop your hypothesis, the research idea, and protocols are developed for actually doing preliminary research through in vitro study. At TL3, now there are three different categories. Like if it is drugs and vaccines, you conduct the initial characterization of the target molecule. If it is device, you are looking at analytical and laboratory testing as a part of physical validation. If it is diagnostics, then you are looking at expression and purification of the diagnostic agent, its characterization, and initial validation at the lab level. Now, when it comes to TRL4 for veterinary products, again, in vaccines, you are working on optimizing the production, right? And then you are looking at purification and biological validation of the molecule. When it comes to devices, you're looking at basic physical components that are integrated to establish that they will work together. That is first laboratory test. And when it comes to the diagnostics, again, you are looking at physical and biological characterization of candidate antigens. So in case of veterinary products, at TRL5, that is early stage validation, again, you are doing limited number of animal studies, especially serological studies. For devices, you are looking at laboratory integration of components where you can get the performance tested. You are looking at all the diagnostic kits to have the desired specificity and sensitivity based on the data generated. And at TRL6, that is again early stage validation. For drugs, you are actually looking at the shelf life of the drug, right? You're conducting the toxicity study. And devices, you are looking at a representative model or your prototype system, which needs to be tested in the relevant environment. And when it comes to diagnostics, you are conducting a lot of field trials at the TRL6. So this is very important. And when it comes to vaccines, you are looking at actually duration of immune response for veterinary vaccines. And at TRL7, that is late stage validation, again, you are working on the GMP manufacturing scale up, doing extensive field trials for these veterinary drugs and vaccines, and confirm sometimes by challenge studies wherever possible. And you are also looking at third-party validation. Most of the times, these are the government agents. There are government agencies who do third-party validations in this sector. For devices, again, the prototype near, that is actual system prototype, is being tested. Sorry. And for diagnostics, again, you have third-party validation to be done by a government agency. And when it comes to pre-commercialization stage, the regulatory approval has to be obtained for new application, and you get commercial manufacturing license for market introduction. And of course, TRL 9, you have commercial launch and post-marketing studies and surveillance also done for the veterinary product. So I was just trying to give how the journey of innovation, right, from ideation to market happens, even in the technologically intensive, cost intensive, time intensive, and science, deep science intensive sector like pharma and healthcare happens. So this gives some idea as to the moment I say where I stand as far as technology, that is product, process, or service readiness is. So TRL1 being ideation, TRL9 being commercialization. So in the pharma and healthcare sector, like incubation center, our incubation center, as all of you know, is helping to do handholding for all the TRLs. So that's is very important that you provide 360-degree support to all these innovators, right from domain expertise, networking, mentorship, collaborations, or any other support, be it legal, IT, marketing, or even supporting for the funding for the researchers to actually go from one TRL to another TRL, right? So this is a elevation from TRL1 till TRL9. So that's what role an incubation center plays. So, oops, this is, so do you have an innovative idea in pharma and healthcare sector? In that case, you can contact ASLMCP and we'll help you for taking your journey from TRL1 to 9. Thank you for kind attention. And I stop sharing the screen. I am not sure. Yeah, stop sharing. If there are any questions, I am happy to take.
Thank you, sir, for such an insightful session. If anyone has questions from the audience, can ask sir, and the link for attendance is shared in the chat box.
It's very difficult to make out whether you got something from this talk or not. I hope at least the slides can give you some information.
We can conclude the session if there are no questions.
It's already 5:45.
Yeah. Gargi,
Yes, sir.
do we have any presentation?
No, sir.
Any certification?
Yes. So I would like to invite Dr. Praful Bharadia, sir, Professor, Department of Pharmaceutics, to felicitate Dr. Savale, sir, with a certificate of presentation.
Thank you, Dr. Savale.
Screen is visible?
Thank you, Dr. Savale. Hello.
Screen is visible, sir. Certificate.
Yeah, it is visible. Okay.
Oh.
Right. I thank you, Dr. Shrinivas Savale, for a wonderful session. And it is an honor to present this certificate of appreciation to you for giving insights into such a difficult topic of understanding how technological readiness levels are distributed and how we can map them. Uh, it is through guidance and mentoring that we are able to go through these technological readiness levels. It's not easy, and therefore, looking at the technical rigor, uh, I thought it was wonderful to have the insights of Dr. Savale to make us understand these technology readiness levels and how they mean whenever we are doing any innovative work. So therefore, looking at the whole presentation, it was very nicely divided into two sections where Dr. Savale has first talked about the technology readiness, the process of innovation and development, and then he has again switched on to the technology readiness level, how creativity and innovation can be used, what is design thinking process, what's the difference between innovation and invention, and then what global innovation index means to us as we are climbing up the ladder slowly, and what's India's position and what are the challenges which you face as you go in the innovation index, and then how the technology readiness levels 1 to 9 are decided, and giving wonderful examples of biosimilars, artificial, uh, you have AI, you have big data, you have veterinary examples for formulation. And with these, Dr. Savale has wonderfully explained the process of innovation development and TRL. So thank you so much, sir, for sparing your time and explaining this process to our faculty and our students. Thank you so much, sir.
It's my privilege and honor as always. Thank you.
Dr. Chabra is also available online. Sir, would you like to add anything? Dr. Chabria? Sir. Sir, you are muted.
Yeah. So thank you, Dr. Bharatiya. Now, Dr. Savale has explained this process very nicely, and I hope all the participants, specifically the students and the faculty, has benefited by this. And I would consider the faculty and the students of LM College of Pharmacy are fortunate that they have all the platforms ready at our institute. So they all should take benefit of this. That's what I want to say. Thank you, Dr. Savale, for a nice presentation and elaborate explaining all the processes nicely.
My privilege, sir. Thank you.
Thank you. Thank you all.
Thank you everyone for joining. And we will now conclude the session and end the session. Thank you for joining.
Thank you.
Thank you, everybody. Thank you, sir.
Thank you, sir.